I'm glad to present our last speaker, Dr. Ben Liesfeld. Ben's career has led to significant contributions in the field of medical technology. After his PhD in physics, he was part of a startup focused on developing cutting-edge ophthalmological medical devices, including computer laser, guided laser surgery systems. In 2013, Ben transitioned to genetic diagnostics. He established the IT and the bioinformatics department at a clinical laboratory for genetic diagnostics. Shortly after, in 2014, and since then, he has been the co-founder and managing director of Limbus Medical Technologies. So let's give a warm welcome to Ben.
Thank you. Thank you, Jimena, for the introduction. I'll stick with my short title. I think it conveys everything that I'm going to talk about today. I'd like to highlight that I'm not only employee of Limbus, I also a special role. I'm also one of the persons responsible for regulatory compliance. That's why I'm going to speak about some regulatory issues regarding AI at the end of my talk as well. We, as a medical device manufacturer, we have always to think about how can we translate, how can we bring these exciting research achievements to the patient, and that means how can we turn it into something, a solution that's going to be approved by a regulatory body. Let me start with a show of hands. We're always interested in getting the opinion of our audience. How many of you, just with a simple show of hands, how many of you have used artificial intelligence at work? I'm sure you, okay, so many hands, yes. I would say the majority, and this is what I would judge as well. So AI has obviously been around for a lot of time, and but the expectations of what AI can do or what AI should do and what we hope for of developments in the future is very diverse. When we ask our audience, what do they want from AI, this gets into a very complicated discussion typically, because the expectations are so diverse. So it starts with, let's just accelerate my daily work. I have very specific questions. For example, is this certain variant pathogenic? Can you assist me here with some additional information? But also the questions become broader and the expectations become higher. Please classify variants for me that I don't have enough information about. Or even, find something that I haven't found so far, or even please establish new gene phenotype associations for me. And you can imagine that all these expectations are very diverse, and I'm going to explain today, I'd like to give a short overview of that all these questions require different approaches, different technologies, and, quite different types of AI to solve them. So that's where I'd like to start with the very basics, just to establish some common terms that we're going to use when we're talking about AI. What types of AI are there? If you look at the very general field of artificial intelligence, one definition is, artificial intelligence is something, it's a machine that you build for a task that you would need human intelligence for. And that means that in this broad definition, a lot of things would fall under this category. Even a calculator could be called an artificial intelligence, but certainly those deterministic algorithms like Exomizer, Phenomizer, our HBO SIM score could be called artificial intelligence in that sense. If you now look at the field a little bit more specific, there's a very important field inside artificial intelligence, which is called machine learning. So it employs certain learning mechanisms to create artificial intelligence. And one of them is probably very well known to you. Your spam filter in your mailbox works with machine learning. You wouldn't be able to survive in your daily business without that spam filter. But also for example, some variant scoring tools like our the virus variant score are based on machine learning algorithms. Now, a very specific field inside machine learning is actually deep learning. sometimes this gets mixed up or the terms are not used in a consistent way. But deep learning is based on deep neural nets, structures that actually mimic how our brains are supposed to work. And for example, face-to-gene, something that's present at this conference as well, is based on that. Alexa, Siri, and of course large language models like ChatGPT. So now that we know kind of what the types of AI are, there are certain common basic processes, basic features that artificial intelligence algorithms have in common. So kind of the basic phases in the life of an AI, how does AI work in general. So you can compare artificial intelligence to an athlete. And the life of an athlete is typically consists 99.9% of training, right? He's training and preparing for kind of the competition in the end for almost all of his life. That's how artificial intelligence works as well. You have a large body of raw training data. You may have to pre-process that data to make it digestible by a machine. And once you have that pre-processed training data, you feed it to your training algorithm and you create model data that you can use later to actually, create new results from new data. But this training is very cumbersome. It's sweat generating. It's expensive. It requires a lot of CPU power. And that's kind of the image of the athlete fits here very well. Then, of course, you need to, you need to check, you need to measure your achievements, you need to validate your model. The athlete needs to measure his progress in training. So you feed validation data, which should be very carefully crafted, very carefully selected. You feed it to your machine learning platform, you apply your machine learning model to it, and you obtain validation results. And like in sports, sometimes there are what we call, with the German term, Trainingsweltmeister, the king of the training ground. So it's not always clear if, how validation was performed, how representative was validation data, maybe the validation data has it already been part of the training data. So good performance in validation needs to be carefully observed and requires a lot of work. And kind of what's the actual performance of the task in the end, this is called inference in artificial intelligence. That's kind of the competition for the athlete. It only takes a few seconds, to make that jump, to run that race. And this is similar for artificial intelligence. You have inference data, you have your patient's case, you have a genomic variant, you feed it, you may have to pre-process the data, then you feed it to your model algorithm, and you obtain a result. And this process is typically pretty fast. That's the advantage of AI. The training takes a lot of time, takes a lot of money and resources, but the inference process, this is fast. And this is faster than other methods that you use, for example, deterministic large simulations, algorithms. This is the benefit of artificial intelligence. Then there are basically two different ways how you can train your artificial intelligence. One of them, supervised training, is probably something that you have all gone through at school. So there is basically a teacher, the developer has a set of exercises that he knows the answers to. And he's feeding those exercises, or he lets the student practice those exercises to make sure that he can reproduce those results. So this is about making the AI learn the relation between input and output. You give it a certain input, and it should reproduce the expected output. In order to be able to do that, you need data that has labels. And the label means that you know the expected answer to. This is not always the case, right? So it depends on your application, on the data you have available, if you can actually perform this kind of training. And there's another kind, or the second kind of training, which is called unsupervised training. And this is rather, feeding the AI a lot of data that has not been labeled, and let the AI figure out correlations, clusters on its own. The benefit is you don't need labeled data, of course, but on the other hand, kind of the result is not really nicely determined, or accuracy is not kind of the goal of the exercise. And there may be, of course, mixtures of these approaches, combinations. And there's not just one single kind, or if you look at a specific solution, it may consist of different building blocks of different kinds of artificial intelligence. A question that we are frequently asked by our customers is, well, you know, kind of it's an intelligent system, right? So it should keep learning. With every case that I feed to it, with every variant that I assess, you know, it should get better, right? And well, we have to calm down our customers a little bit. You know, there's a very good reason to stop learning, actually. And well, the first reason that I already explained is efficiency, right? The training is so expensive, I don't want to do this every day. This is just, yeah, it could be wasting money, or it could be at least very expensive to do that. Well, the second very good reason is generalization. So you actually designed your artificial intelligence, you designed your training data to be representative of, you know, the entire world, all potential patients, and you trained your AI, you know, to be able to translate this to every other new patient, to every other new information that it's encountering, and still produce a good result. If you're now feeding your artificial intelligence data from your daily work, probably mostly white Caucasian patients, you know, you're losing on this generalization, and you're probably gearing your artificial intelligence towards a certain bias, and we don't want that. So again, you know, kind of the training is very cumbersome and needs to be carefully designed and should not be changed, you know, just without good oversight, without good design. And last but not least, there's a matter of reproducibility. We are in the medical discipline. We would like, you know, a question that we ask the system to be the same today or tomorrow. If this could change, you know, without control, this could be an issue. This is certainly a regulatory issue. So we need systems to be consistent and not change over time, and there should be at least, you know, very controlled steps when the artificial intelligence can further develop. So let's take a look, with all this in mind, take a look at some use cases in genetics and how we could address them, how we could use these tools for that. So I mentioned, you know, the use case, you simply accelerate easy cases for me. That would be, you know, training it on known variants. These are the variants. This is the expected outcome. So this is certainly a supervised training, very straightforward. Well, if you think about your training in AI in identifying negative cases, this is probably a little bit more difficult. At least you need cases with high confidence that these are negative. You need to validate that it performs well. Still, a supervised training. If you think about automatically classifying variants, well, one could argue that you don't even need AI for that. Even ACMG 4.0 is still a nicely rule-based system, but you could think, yeah, typically I don't have all the information available. Couldn't an AI, you know, just supplement information that is probable? Well, you could train an AI with that, but still it would be supervised because you would train it on known variants. They are solvable, but difficult cases. This is very interesting, and we know that there's a significant number of patients that don't suffer just from one disease, who have overlapping phenotypes, multimorbidity. This is certainly something that where AI can help. That could be a unsupervised approach. So let the AI try to cluster phenotypic features and genes and how they may correlate. If you are trying to get more variants of unknown significance to a higher level, that might be suitable for a supervised approach. Yeah, we also heard that people would like AI to write the reports. Well, this could be just a ChatGPT version that, you know, summarizes for you some abstracts. But for example, which could be an unsupervised training approach, but if you need more accurate results, you need to improve on that. So you need to at least specialize that general model into something that achieves a higher accuracy. And I'm going to give you a few examples on the next slides. And of course, there's this very challenging task of generating new gene phenotype associations. And of course, this is certainly dependent on the information that we have available. The AI is not a wizard. It needs something to build upon, but that would also be an unsupervised approach. So let's take a look. We all know we have all used, or many of us have used AI in the field. What has been achieved so far? Well, I've been a little lazy. I gave ChatGPT the task, please create a table of bioinformatics scores that use machine learning approaches with their publication year, and just create a nice looking table of it. It did so, but there were so many mistakes and errors in this table, I can't show that to you, so I had to do it by hand. I didn't go back further than 2010, but it's obvious that we have been using machine learning in classifying variants for a long time. It's been proven very useful, and everyone who has been classifying variants in the past has used those algorithms. So what's new now? Why the fuss? Why the excitement as well about the new achievements? And to explain this a little bit, let me take an example of totally outside our field, weather forecast. My background is I'm a physicist, and the dream of physicists is, let's put all the physical formula, the rules that we know how the world works, let's put this into a computer and feed the computer with a good representation of the current state of the world, and then we can predict any future state of the world with high accuracy. And we even built supercomputers called Earth Simulator for that. And but it turned out that this is very difficult, and even for doing something so simple like a weather forecast, we need huge machines, takes a long time to calculate, and these models are very difficult to develop. Now comes along the DeepMind team from Google and applies a deep neural network to the weather forecast. And this is particularly disappointing because this model, this artificial intelligence, knows nothing about physics, about the weather. It doesn't know that heat goes up and that rain falls down. It is totally ignorant of that. But the sad story is, or the bitter lesson for us physicists is, it works better than the carefully crafted system where we put so much of our human knowledge into. It's just brute force analysis of statistical patterns. And it doesn't even need so much information. You train this statistical model and it can now predict the weather in 10 days ahead with acceptable accuracy, and can do that fast. So you can run your own weather forecast on a laptop computer, and this is, obviously this is a great advantage and advancement, but it also puts, all those expensive solutions that we had so far in a corner. Now, of course, this has also been applied to biology, to genetics, protein folding, for example, AlphaFold, also from the DeepMind team. It took us decades, many lives of scientists to discover those protein structures from about 100 molecules. It's extremely expensive X-ray crystallography, and we feed this to a deep neural network, and it can now predict protein structures from unknown proteins with extremely high accuracy. So we probably don't need all this expensive science anymore. And of course, it's a small step from protein folding to predicting what is, let's take a small change in my DNA sequence, what kind of effect does it have on my protein? And of course, this was also done by the DeepMind team. And it delivers results, at least for missense variants and the coding sequence. It provides a very good predictor of how the protein is being folded and what kind of impact it has on the organism. And now, just a couple of weeks ago, AlphaFold 3 has been published. And this is really exciting because they don't only consider proteins, they consider all kinds of molecules in the living system. And they can, for example, now very accurately predict binding sites of transcription factors. So imagine what this can mean for all the non-coding regions that we're looking at with whole genome sequencing. This is something I recommend everyone to monitor. Just as an example of how large language models can play a role in genetics, and I know that some companies have developed or have modified language models. I just want to highlight with one example how this can lead to unexpected results. I was feeding that specified or this specialized AI, specialized for genetics. I fed it a question about BRAF V600E, a well-known variant. I think I don't have to explain it to you. Well, for this, asking for this variant, I got a reasonable result, which from my point of view did not contain any errors. Well, then I asked it about a different variant, seemingly different variant, and was curious about what the result would be. And then kind of the answer gets really misleading. It just generates false results. And if I really ask it specifically about the difference between those two variants, it turns out it has some knowledge about that. So it knows that these are related and that one is older, or the older description, one is the newer, but it mixes up which is which. So in the end, we have to say, well, large language models may be helpful for specific tasks. In genetics, if you're looking for, or if you're relying on your tools, if you're looking for accuracy, you have to be very careful. These models don't have a concept of a genomic variant. They just don't know what it is. They don't even have a consistent world model. So they don't know that two different descriptions of a variant could mean the same genomic variant. And they somehow, they have some knowledge about it. I mean, they have been fed scientific literature from humans, but they don't know how to deal with inconsistencies or how to apply that knowledge. And now imagine, this was a very prominent variant. Imagine some more complicated cases that are not so well known. Keep a critical mind when you deal with output from those large language models. This needs additional supervision. And some coined it even, the statistical parrot. These large language models simply are very good at predicting what the next word should be. And the critical minds can say, we all are statistical parrots, we are neural networks, but we have additional methods to control our output and just don't take this output as is and check it for accuracy. We don't even need to go this detour about the human language. Why should we analyze our inconsistent, misleading scientific literature? Let's just feed the genomic sequence to these large language models and see what they can do with that. There are some very exciting projects about this, but I don't have the time to go into more detail here. So how much time do I have left? Okay, so I have a few minutes to touch on the regulatory aspect. What is actually allowed? What can we do? We're dealing with patients. We cannot just do anything. There are two important aspects regarding regulation. One is, of course, the regulation that should be applied, that must be applied to medical devices, the medical device regulation or the in-vitro diagnostic regulation in the European Union. And just recently, the AI Act has passed, so it's already in force. And these two regulations provide basically the framework that we have to work in. If you look at how devices are classified in these two regulations, I think Jimena did a good job explaining to you how these risk classifications work in IVDR. And with genetic testing, we are very clearly Class C and in Class C devices. And the AI Act has a similar risk-based classification. It starts with low risk, limited risk, high risk. It also has a classification of prohibited devices, very curiously. And it's also clearly written in the regulation that Class C devices that are already subject to conformity assessment by notified bodies have to be high risk, have to be classified as high risk devices. We have basically 36 months as manufacturers and regulatory bodies to implement all these new rules. This will be challenging, but at least kind of the rules for this play are clear, and this is something that we can work with. And actually, for us medical device manufacturers, it's not so much new. On the left side here, I displayed these are requirements contained in the AI Act that we already know, that we already subject to due to our being a medical device, so conformity assessment, performance evaluation and such. Something that's new in the AI Act is actually that we need to enable human oversight. That's something new. And probably kind of one of the solutions for that is probably designing AI such that it can explain how it came up with the result. This seems like a necessary feature according to the current regulation. It may be, however, difficult to implement depending on your choice of technology. So it's more difficult when you're using deep neural nets to explain how it came up with that result compared to other machine learning approaches. But that's basically details. That's something that the developers of AI have to deal with. Well, so finally, I think I get easily excited about those research advancements. I think there's a lot of things to discover. We know so little about the DNA. AI can help, but can also make mistakes, just looking at this picture. But we'll follow the news, and we are going to try to develop solutions that are accurate enough for clinical diagnostics. And now I'm happy to take your questions. Thank you.